The last topic we're going to talk about in Chapter 6 is RNA interference, sometimes called RNAi therapy. What this is referring to is downregulation of gene expression through the use of small RNA molecules. In other words, when a gene is made into messenger RNA, made into enzymes, you can control how much enzyme is being made either by inhibiting translation of the RNA or by prematurely destroying the RNA by these RNAi interference strategies. Now, let me stress before we talk about how it can be used in medicine, RNA interference actually is a normal process. That you and I have RNA pathways in our body right now, and we use it in defending cells against viruses and in the issue of these transposons that's often talked about in virology portion of microbiology. But here we're going to talk about it in terms of what's going on in the biochemistry of RNA interference and then how is it being used. In the biochemistry of the RNAi pathway, there's a very long double-stranded RNA to which an enzyme called dicer. The dicer enzyme will see this long double-stranded RNA and cut it up into fragments. And these fragments are called small interfering RNAs, siRNA. And these siRNAs, very small, they're about 20 nucleotides in length. And what happens is they'll start to unwind in the presence of a protein complex called RISC. So the RISC complex of proteins will now start to unwind the siRNAs and generating two strands. On the left side, one of the strands is called the passenger strand of the RNA, also called the sense strand. And the passenger strand is degraded. Forget about the rest of it. Not important. It's just degraded. On the right side, the other strand is called the antisense strand, sometimes called the guide strand, to which the RISC is still bound. So RISC is bound to the guide strand in the cytoplasm. Meanwhile, in the nucleus, there is a gene that's making RNA, and then the RNA comes out into the cytoplasm. And the RNA is supposed to encode for a protein. But now if the guide strand is present, the guide strand, because it's complementary, will base pair in a complementary anti-parallel fashion to the messenger RNA. And then in the RISC, there's a protein called argonaute. An argonaute portion of the RISC will then degrade the messenger RNA by nuclease action, and now the messenger RNA is degraded, and when the messenger RNA is degraded, the encoded protein of the RNA cannot be made, and so therefore you're downregulating gene expression. The gene is made in the RNA, but you cannot make protein because the messenger RNA for the protein is degraded. So in this RNAi process, the result is post-transcriptional gene silencing. So even though the gene is made, even though it's making messenger RNA, you're effectively silencing the expression of the gene. And this is referred to as knockdown. You're knocking down the expression of the gene. This is in contrast to another term called knockout, where the gene itself is disrupted. So in gene knockout, by inserting nucleotides in the gene in a chromosome, the gene is effectively useless. That's called knocking out of a gene. But knockdown means the gene is present. Gene's making messenger RNA. The gene's doing what it should be doing, making messenger RNA. But the RNA is knocked down and therefore reducing gene expression into the protein being made. So these words, knock down versus knock out, please make sure you know the distinguishing features of the two. Knockout, you're knocking out the gene. Knock down, you're knocking out the messenger RNA being made from the gene. Regarding RNAi in therapeutics, RNA has a very short half-life because RNA is often broken down, degraded by ribonucleases. And to time-limit the effects of RNAi, what can happen is siRNAs can be encapsulated in lipid vesicles, administered, or can be conjugated with adducts to try to get the siRNAs into cells to knock down gene expression. A single RNAi treatment can silence gene expression for up to 2 weeks. That's pretty good if you think about it, because RNA is degraded very rapidly, and by conjugating the RNAi, you can get that type of therapy to last up to 2 weeks, knocking down gene expression. And regarding if they were to ask on board exams, how can you have the RNA stick around longer? Because on 2' position of the ribose, the 2' hydroxyl can be replaced with either a methyl or a fluoride group or fluorine group. And by doing so, the ribonucleases cannot break down the RNA properly. And so the RNA can stick around longer, and the therapeutic effects can now last up to 2 weeks. And nowadays, the RNAi in therapeutics can be used in the treatment of certain cancers and many neurodegenerative diseases. It can also be used as antiviral therapies. That can be discussed in virology in the microbiology portion of this course.